All right, now, hello everyone. We are ready to begin our third and last lecture video as part of chapter 10, which is all about membrane transport. And in this video, we are going to be talking more about active transport. So in this chapter, we have already covered in the first few videos some information about the thermodynamics of transport from a conceptual point of view. We then moved on to discuss a lot about different types of passive mediated transport. And now we are going to, in this video, discuss a few different types of active transport. So we'll discuss primary active transport in the form of our sodium-potassium pump, as well as our ABC transporters. And then we'll talk about a couple of different secondary active transporters. So first, let's review what is active transport doing and why might we want it. So active transport is the movement of molecules against their gradient. And we call it active transport because we need an input of some kind of energy. So often this is through the hydrolysis of ATP directly. And we can also indirectly use the hydrolysis of ATP for this, as we'll see in our secondary active transport. So just as a little reminder, we talked about a lot in our last lecture about a particular active transporter that's important for the establishment of the electrochemical potential gradient in a neuron. And hopefully you'll recall, this one is our sodium-potassium transporter, which is also called the sodium-potassium ATPase, which is also called the sodium-potassium pumps. Sodium-potassium pump. Three names, same protein. So this is a type of, and this uses a type of primary active transport because this pump uses the hydrolysis of ATP, and it directly is going to provide energy to help drive that transport. We also have, like I mentioned, secondary active transport, where ATP hydrolysis is used to generate a gradient, and then we dissipate that gradient to help drive transport of a second molecule, hence the terminology secondary active transport. So primary active transport is directly using ATP, secondary active transport is indirectly using ATP. So let's talk about a few different types of primary active transport. So one type is a P-type ATPase. These ATPases hydrolyze ATP, and they also transport cations, which are positively charged ions, hence the name P-type ATPase, hydrolysis of ATP for transporting of positive ions. And an example of this that we've already talked about is the sodium-potassium ATPase, or the sodium-potassium pump. Another type of ATPase is the A-type ATPase. These transporters hydrolyze ATP, but instead of moving cations, they move anions, or hence the name A-type ATPase. We also have ABC transporters. These ABC transporters contain an ATP binding cassette, hence the name ABC. And an example of this that we'll discuss in this lecture is called P-glycoprotein. And then we have another couple of types of ATPases. These are actually used for the synthesis of ATPase, and they also take advantage of a gradient. And so one type is the F-type ATPase. These ATPases synthesize ATP, and they use a proton gradient, and these ones are found in mitochondria and bacteria. There's also another similar one called ATP synthase that we'll discuss more later on in chapter 18 in our own types of cells that helps us make ATP as well. And then another type of ATPase that helps to synthesize ATP is the V-type ATPase. This is one that we find in plants. It also uses a proton gradient, and we see this in lysosomes as well as in plant vacuoles. So we have the V there, and that's for the V-type ATPase. All right, so now that we've talked about a number of different types of primary active transport, let's talk more about the sodium-potassium pump. So the sodium-potassium pump helps to establish those sodium and potassium gradients in our cell that allow for our nerve impulses. And just as a reminder, it pumps potassium in and it pumps sodium out. And it does this while it's hydrolyzing ATP. And it's able to carry out this function partially because of its structure, which consists of, in particular, the alpha subunit that we're considering here, which is drawn in rainbow on the side of the slide. It has 4 different domains. So the domain labeled M is the membrane binding domain. So this part is in the actual membrane of the cell. So on the bottom is the extracellular part that extends out the outside of the cell. We have the cytosolic portion that extends towards the inside of the cell. The membrane portion is where we have a lot of our sodium and potassium binding, and you can see where that happens right in the middle. The next domain we'll consider is the N domain, or the nucleotide binding domain, which is shown here at the top. We don't actually have a picture of ATP bound to this domain, but this is where ATP would bind, and it's going to help to propagate some structural changes, and it's going to be helped out by that by the A domain, the actuator, which also helps to propagate some of those structural changes, along with the P domain, the phosphorylation domain, which involves an aspartate residue that is going to bind to a phosphate group. And so the hydrolysis of ATP and the binding and unbinding of these phosphate groups help to make these conformational changes, especially in this region of the sodium-potassium pump, that help it to be open to either the cytosolic side or the extracellular side, where it can help then to pump, again, our sodium out of the cell and our potassium into the cell. And so just as a summary of that, the entire structure as a whole has two main conformations, and we call them E1 and E2. So let's talk about more of the details of that pumping mechanism. So we can start here where it says number 1, we have our sodium binding. And the sodium is able to bind when the pump is in the E1 conformation and it's bound to an ATP. So when you have E1 with ATP bound and the 3 sodium ions bound, we then have the ability to hydrolyze that ATP, and it's going to be hydrolyzed into ADP plus a phosphate. And that phosphate, so the ATP, I want to say, is released just to the inside of the cell. The phosphate now forms this high energy aspartyl phosphate intermediate in that P domain we were talking about before. And then this high energy bond helps to drive this transportation of the sodium to the outside of the cell. And this is going to change the whole conformation of the pump, right? So in the E1 form, the pump was open to the inside of the cell. Now the conformation has changed. It's in the E2 form, so it's open to the outside of the cell, and that's what allows the sodium to leave. Once the sodium has left, potassium can now enter into the sodium-potassium pump. And then we have now the E2 form, which is still bound to phosphate, and now it's also bound to potassium. And at this point, we have the hydrolysis of this high-energy phosphate bond. And so now we have simply the potassium bound to the E2 form of the pump. And then, once again, this hydrolysis of the phosphate is going to drive this conformational change back from E2 back to E1. Once we have this conformational change, now the pump, instead of being open to the outside, is now open to the inside of the cell again. Once the inside of the cell is open, now those potassium ions can be released to the inside, and then a brand new ATP molecule from the inside of the cell can then bind to the pump, and now we're back where we started, where the pump is in the E1 form. It's open to the inside of the cell, and it has an ATP bound, and it's ready to, once again, throw some sodium ions out of the cell and bring some potassium ions into the cell while it's hydrolyzing ATP. So as a summary of this sort of complicated mechanism of pumping, let's go through it again, this time with words. So first we have the pump is in the E1 form. It's going to bind to ATP as well as 3 sodium ions, and it's going to be open to the inside of the cell. That ATP is going to lose a phosphate. It's going to be transferred onto the E1 form of the pump, and that's going to allow for the release of ADP, the rest of that molecule. Once you have this high energy phosphate bond that we created, the whole pump is going to change its conformation from E1 to E2. And then once it changes conformation, now the 3 sodium ions can be released outside of the cell. And so since now the protein's in the E2 form, it's going to bind to potassium ions outside of the cell. And at this point, that phosphate group, the bond is hydrolyzed, the phosphate is released, and the E2 protein is going to change its conformation once again back to the E1 form. So now it's facing the inside of the cell. This is going to let the potassium ions be released, as well as now the pump is in the E1 form, so it's once again ready to bind a new ATP molecule. So overall, we have 2 potassium ions that were pumped into the cell, 3 sodium ions that were pumped out of the cell, and 1 ATP molecule was hydrolyzed. for each sort of cycle of this pumping mechanism. So let's now move away from the sodium-potassium pump and talk about a different type of primary active transport. And we're going to consider here the P-glycoprotein, which is a type of ABC transporter. So ABC transporters, in general, as a class, are able to pump a wide variety of both polar and nonpolar molecules, both in and out of cells. And so in general, ABC transporters have two cytoplasmic nucleotide binding domains, which are labeled here NBD for nucleotide binding domain, right? NBD. These nucleotide binding domains are also called ATP binding cassettes, or ABCs. ABC. So ATP binding cassette, A, B, and C. And this is on the cytoplasmic side. Here we have our actual membrane region. And then here we have the outside of our cell. So P-glycoprotein is a type of ABC transporter, and its job is to pump amphiphilic molecules, which include drugs, out of cells. And when P-glycoprotein is overexpressed on the surface of a particular cell, it can contribute to drug resistance. And this can sometimes be bad, right? So if some cancer cells, they'll overexpress P-glycoprotein on their surface. And that's going to let these cancer cells specifically pump out drugs that might try to kill the cell, right? For example, like chemotherapy drugs. Another example of overexpression of ABC transporters is something we'll see in bacteria, and it can contribute to antibiotic resistance in bacteria. So if antibiotic drugs are trying to get into the bacterial cell, an ABC transporter on the surface of the bacterial cell can then pump that antibiotic right back out of the bacterial cell, and therefore the antibiotic cannot kill the bacterial cell. So how do ABC transporters actually work to transport molecules out of the cell? So first we'll consider an amphiphilic molecule, which is shown here in pink. So it's trying to get into the cell. It might even make it into the membrane or somewhere inside of the cell. But if it's near a P-glycoprotein or another ABC transporter, this amphiphilic molecule is going to be able to bind to some of the hydrophobic residues that we see in the interior of the P-glycoprotein in this region that's spanning the membrane. So just a reminder, this is the outside of the cell on top. We have the cytoplasm shown here on the bottom. So at this point, once we have some kind of amphiphilic molecule bound somewhere on these hydrophobic residues on the inside of the P-glycoprotein, now ATP can come in from the inside of the cell on the cytoplasmic side, and it's going to bind at those nucleotide binding domains 1 and 2. This binding of ATP is going to trigger a conformational change in the ABC transporter, in which these two kind of arms are going to start moving toward each other. And then once they have moved towards each other, that sort of movement of that bottom part in is going to help to move the top part sort of away from each other and create an opening that faces the outside of the cell. And once that part of the cell is open facing the outside, now this amphiphilic molecule is going to leave and go back out to the outside of the cell. And then to return the ABC transporter back to its original conformation, which we saw on the left, these ATP molecules that are bound to those nucleotide binding domains are going to be hydrolyzed. Once they're hydrolyzed, this is going to return the P-glycoprotein or other ABC transporter back to its original conformation. So those were two examples of primary active transport, right? We had our sodium-potassium pump and then our ABC transporter, and our specific example of that was the P-glycoprotein. Now we're going to change gears a little bit and look at a couple of examples of secondary active transport. And so just as a reminder, secondary active transport is indirectly using this ATP hydrolysis. So in this case, we're looking at our sodium and glucose symport. So we're looking at the movement of two types of molecules, glucose and sodium. They are both going to enter through this symporter into an epithelial cell, that's lining your intestines. So you can imagine this was important. For example, after you eat a meal, some of the nutrients, including glucose, are going to be hanging out in your intestinal lumen, part of your GI tract. And you're going to need to take up nutrients like glucose into those epithelial cells that line your intestines, where they can ultimately be sent to the bloodstream and to other tissues where they can be broken down and used for energy. And so the way we bring glucose into these epithelial cells is through secondary active transport, or SYMPort. And so we talked about how that is driven by first using ATP hydrolysis to create a different gradient, and then dissipating that gradient to pull a second molecule across. So let's look at how that works here. So in this case, we have our sodium-potassium ATPase, as drawn here on the other side. Sodium-potassium ATPase, as we already know, is working very hard to pump sodium out of the cell, which creates this very nice sodium gradient where we have lots of sodium outside of the cell and not very much sodium inside of the cell. And so in the SYMPorter, the sodium is going to be happy to travel down its gradient. And at the same time, it's going to bring with it the glucose molecule. So this energy that's being sort of recovered by dissipating the sodium gradient is going to be what helps glucose also travel through this SYMPorter along with the sodium. And once the SYMPorter has worked, now since we have this sodium that has gone down this gradient, we still have our sodium-potassium pump over here to help recreate that gradient if we ever end up essentially sort of equilibrating the sodium inside and outside the cell. That we don't let that happen because our sodium-potassium transporters are always working to pump that sodium right back out, which we see here, as well as pushing potassium back in. And the other thing to note here is once the glucose has gone into the epithelial cell that's lining your intestine, it then gets actually pumped right back out into your capillaries, into your bloodstream, where it can get delivered to other systems of your body. And this is done using a uniporter. So that's similar to the passive mediated glucose transport that we discussed in our last lecture. In that lecture, we talked about GLUT1, which is found on the surface of red blood cells. In this case, we have a different glucose uniporter. It's called GLUT2. And so this one does not require energy. It's just going to allow glucose to travel down its gradient into the bloodstream. So essentially, once you have a high enough concentration of glucose inside of this intestinal cell, that helps to create a gradient where you have high glucose inside and low glucose on the other side. And then the glucose can travel right down the gradient through this little uniporter. And then the other thing I just want to emphasize here is the terminology. So here we have our sodium glucose SYMPorter, SYM meaning same. So sodium and glucose are both traveling in the same direction. Our sodium, in contrast, we have our sodium potassium pump, which we saw on the right side of the cell over here. This one is using antiport because sodium is traveling out, but potassium is traveling in. So when you have two molecules traveling in opposite directions through a transporter, that's antiport. If you have two molecules moving in the same direction through a transporter, that is SYMPort. And then sometimes you'll have direct hydrolysis of ATP driving this movement, such as in our sodium-potassium pump. You'll also have sort of that indirect uses of ATP, which we see in our sodium-glucose SYMPorter, which uses the sodium gradient that we created with our sodium-potassium pump. So that's indirectly using that ATP to help drive that glucose movement. All right, so let's look at another example of secondary active transport, and this one is going to be lactose permease. And this particular pump is found in bacteria, and it helps to pump lactose into bacterial cells from the outside because, similar to glucose, lactose is a type of a type of sugar, not monosaccharide, that can be used for energy by the bacteria. So here, just to orient you, once again, we have the periplasm. This is towards the outside of the bacterial cell. And then we have the cytoplasm, that's the inside. So in the previous slide, when we talked about our sodium-glucose SYMPorter, that was driven by a pre-existing gradient of sodium ions that was created by the sodium-potassium pump. Here we're using a different pre-existing gradient of protons, and we see that here with our proton pump. So through a process of oxidative metabolism, bacteria are able to use this proton pump to drive positively charged protons out of the cell, which creates a proton gradient, where you have high concentration of protons on the outside of the cell and a low concentration of protons inside of the cell. And so we're going to see a similar idea of symport, where the protons now are going to be able to move down their gradient, and they're going to bring the lactose along with them, similar to on the last slide where we saw sodium moving down its gradient and bringing the glucose along with it. So let's get more into how that works. So our lactose permease pump also has this E1 and E2 conformation, just like we saw with our sodium-potassium pump. When it's in the E2 conformation, it's open towards the outside of the cell, and it's going to be available to take up a proton and bind a proton. And then once the E2 form has bound a proton, it's also able to take up a lactose from outside the cell in the environment. And then now we have this form of the E2 with a proton bound as well as a lactose bound. And then we have the actual transport, and this transport is driven once again by that energy gradient of the proton moving down its gradient, and it's bringing the lactose along with it. At this point, and this is going to help to drive this conformational change, where the pump goes from the E2 conformation to the E1 conformation, and it's open to the inside of the cell. At this point, once it's open to the inside, the lactose gets released, and the proton gets released. And then the E1 form can recover and retake the E2 conformation. And while this recovery is happening, we also have the proton pump working hard to maintain that proton gradient. So we dumped all these protons inside the cell, or one proton for each cycle inside the cell as we move the lactose in. That proton pump can help reestablish that proton gradient by pushing the protons back out of the cell. So with that, now we've covered a number of different types of active transport, and you're ready to answer the Chapter 10 active transport practice questions.